Recording in progress.
Well, a very good morning to all of you. Township. So good morning to all of you. Today I am going to present my work that is on fish morphology. This video lecture will cover fish morphology and their morphometric technique. So first of all, while starting with the topic, first of all, I would like to introduce the topic. While I am saying fish morphology, what is the importance of fish morphology? In almost all cases of fisheries sciences, there is a basic requirement what type of fishes you are working on, what is the variety of fish. Okay, so in almost all cases of scientific inquiry, whether theoretical or applied, there is a proper identification of species is required. So the basic criteria of identification is based on the morphological features, that is on traditional morphometry. If traditional morphometry is failed, then you can go through the molecular techniques. So firstly, I'm going to describe fish morphology. So myself, Dr. Nisha Rana, I'm assistant professor in Department of Geology, Kerala Verma Subharti College of Science. Additionally, I'm also working in Central Research and Incubation Center, Swami Vivekananda Subharti University, Meerut. So I'm starting with the topic that is the fish morphology. These are the content of the topic, that is the introduction, importance of the morphology of fish. Morphology of fishes, what type of morphology fishes are having, and their identification keys on the which basis we can identify what type of fishes. The fin formula, that is another morphometric feature, and what is the importance of fish taxonomy and the references. Those I wish to say. So first of all, starting with the introductions of the topic, as we know, fishes constitute an economically important group of vertebrate and also found in almost all major aquatic habitats. And fish identification is traditionally based on external morphological features, their body shape, size, and the coloration also. The fish can show an astonishing diversity of shape, size, and color. So when I am talking about the importance of morphology, you can see the many type of fishes on your screen. There are so many variety of fishes and so many species of fishes, but you can see their shape may be different, their color may be different, their morphometry may be different, starting with salt, reddish fish, and large spotted dogfish, pike, sea bram, eels, herrings, gurnards, swordfish. These all are having different morphology and of also different colorations. So how can we identify a fish? These are the morphological features of a fish. That is the Mistus cavaceus. Mistus is the genre and the cavaceus is the species. You can see an image of fish showing envy. Envy is the nasal barbels. The presence of barbels may be vary. There may be mandibulary barbel. This is showing MB, OMB, IMB. Okay, this is the mandibular barbel. Those are mandibular and outer mandibular. Okay, so there may be a lateral line system that will be definitely present there. The lateral line system starting from the base of the operculum and the starting point of the caudal peduncle. So the labeling of this is the labeling of a siluriid fish, that is a non-scaly fish. Non-scaly doesn't mean that the fish don't have scales. Scales are embedded in the skin. So starting with the fins. Fins may be various types of fins. The first will be the dorsal fin. The maybe adipose is present or may be absent. The caudal fin. The shape may be vary. The deeply formed maybe heterocircle, more circle. The shaped size structure may be vary according to the species. The fin may be pectoral, ventral, anal, and then the caudal. The fin ray count, that is also another morphological feature and also morphometrical feature. The number of ray count may be vary according to the species, that the number of ray and number of count will be fixed in a particular type of fish species. So this is a scaly fish showing the morphological features and the morphometry of the fish. The fish name is Nandus. Nandus is the genre and also Nandus is the species. The ham dot denotes that the fish was described by the scientist Hamilton. Okay, this is a scaly fish. Although all scales are not mentioned on the skins, are denote and these are the various types of fins, and the labeling starting from A to H is a various morphometrical feature. For example, when I am starting with A to H, that is the total length of a fish. Total length starting from the tip of the snout, from the last base of the caudal fin. Okay. Then the A to B. A to B is the snout length. This is the known as snout length of a fish. That is also a morphological feature, not only morphological, morphometrical features that will be used in the morphometry of fish. And the B to C mark denotes the distance, denote IOB, that is the interorbital width. Okay, you can see the labeling below the figure. Everything is mentioned on this image, and the A to D, this is the total snout starting from the base of the snout to the base starting point of the first dorsal fin. Okay, the dorsal fin in this fish is in R2, first dorsal fin, and then the second dorsal fin, and these are the spiny rays of the first dorsal fin. You can identify we can mention the same in the fin formula. On the basis of spines, you can also identify the fishes, but the number of fin rays and the number of counts and the number of soft rays will be fixed in a particular type of fish species. Okay, then the I to J is showing the depth of the body, and this is the caudal peduncle, and A to G is the standard length of a fish. Okay, that is premaxillary, mandibularies, okay, maxillaries, and barbels may be present in a particular type of fish species and may be absent. There the number of barbels in a particular type of fish will be fixed. Okay, these are the fish fin rays. These are the pectoral fin rays. The number of pectoral fin rays in all Nandus will be fixed. The number of pelvic fin rays will be fixed. Anal also, anal one, and then the caudal also and dorsal also. So these are the prominent characteristic fishes in a particular type of species. So this is again a showing a morphology of scaly fish, showing various morphological feature. Those I have already described, the snout length. The last image showing A to H accordingly the labeling, and this image is showing the particular labeling of same, the snout length, nostril premaxillary, mandibles, maxillary, then the pre-opercular, super-opercular, pectoral fin, then the dorsal fin, and then the spines, then the soft ray of the dorsal fin spine. You can identify a particular type of fishes by using, you can touch the spine tip. If it is spiny, then you can count the on the spine. If it is not spiny, it is in soft form, then you can say these are the soft fin rays. Okay, this is the total body depth of a fish. There may be adipose present in a particular type of fish. It is not mandatory that it will be present in all fishes, but it may be present, it may be absent, but in a particular type of species it will be present or it will be absent, but the characteristic feature will be fixed in a particular species. There is another important feature, that is a lateral line system, that is starting from the base of the operculum and the starting point of the caudal fin. The number of scales in a lateral line will be fixed in a particular type of fish, and it will be a prominent characteristic feature. Lateral line may be single, may be double dotted. It may vary according to the species, and there will be a lateral transverse row that is starting from scales above the lateral line, scales below the lateral line. You can identify and count 1, 2, 3, 4, 5, 6, 7, and then 8. And it will be count as half because it is on the center of a lateral line. Then scales below the lateral line, you can same kind count by using same method: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So these are the prominent characteristic morphometrical features. So you can identify on the basis of gill arch that this is a normal, generalized normal gill arch. This is the pharyngeal teeth of surf perch. This is the pharyngeal teeth of carp, yani ki Cyprinidae. As you know, Cyprinidae is the largest family of fishes. So this is an image of a fish showing the exotic fish. I have mentioned the exotic fish. This is a fish, Cyprinus carpio. You can see the morphological feature of the Cyprinus carpio is totally different in from another species. This is a silver carp. This is the grass carp, Tinopteranodon idella. So you can see starting from first fish, second, silver carp, and grass carp. All these are the carps, but their morphological features, their fin shapes, their body shape, their fin number count will be different in each and every species. So there is another feature that is known as fin formula. There is a fin formula. You can, by using the fin formula, you can count the number of fins, number of scales, and also identify the fish. For example, I have mentioned D18 to 19. It means the number of rays in dorsal fin will be 18 to 19. It may be 3 by 15, 3 by 15 showing, 3/15 showing that 3 will be spine and 15 to 16 will be the soft fin rays. P9 denotes pectoral fin rays. These are all soft fin rays will be 19. Ventral lines showing ventral fin rays will be 9, and the anal A showing A dot. It's showing that anal fin rays will be 8 in total, but 3/5 is showing that 3 will be spine and 5 will be soft fin rays. Caudal 19 showing the caudal fin rays will be 19. LL43 means to say lateral line scale. It means the number of scales on lateral line will be 43. If I'm talking about LTR, LTR means to say lateral transverse row. Lateral transverse row will be 7.5 upon 7.5 dash 6.5. 7.5, it means scales above the lateral line, and 6.5 means to say scales below the lateral line. And then half and half is showing that the one scale is on the lateral line, that will be also a countable number. If I am talking about the number of barbels, that it is mentioned the barbels number will be one pair. It means in a particular type of fish species, the number of barbels will be one pair. Okay, these will be always in the pairs. This is the fin formula of katla katla. By using scales and divider, you can do the morphometry of particular type of fish. If I'm talking about the shape, you can see the shape of lower lip, upper lip, that will be a prominent, and the lower lip also cover the upper lips. Eyes, eye orbital, you can measure by using divider. Okay, and you can also count the number of scales on lateral line while you are having a fish and doing the morphology. The fin formula of this particular species of katla will be fixed. So when I'm saying katla katla, katla katla means to say the katla is the gender and the species name also the katla. Okay, and the fin formula that I have mentioned below the figure is the fin formula of a particular type of fish. So this is the fin formula of Sirenus mirgala, that is different from the previous one, but it denotes that the number of scales, number of fin rays, number of soft rays will be fixed in this particular type of species. It means in case of all mirgala, the number of fin rays, dorsal, pectoral, anal, caudal, lateral line number of scales of latent transfer, so will be fixed in all Sirenus mirgala. Now, next is the Labiorohita, also known as Rohu. These are the major carps, Katla Katla, Labiorohita, and Sirhinus Mirgala. Okay, you can see the mouth is fringed. That is another morphological feature of this particular type of fish species. Labio is the genre, Rohita is the species of this genre, and the fin formula for this species is this one. So I have mentioned a comparative chart of four different type of fish species of Chandra. Generally, locally, this species is also known as Soli. But in case of Soli, there are four locally found species in Western Uttar Pradesh. If I'm talking of Western Uttar Pradesh, that is the Channa Gachua, Channa punctatus, Channa stratus, Channa marulius. You can see the all fishes, are commonly known as snake-headed fishes because they are because of the appearance of the head region like a snake. Okay, but the variation in their morphological features, the fin may be same but same type, but the number of rays in a particular type of species will be fixed. You can see the punctatus body, you can see the stratus body, there may be stripes on the lower base of the body. You can see the marulius, there may be a black blotch, black dot on the lower surface of the body. So the morphological feature, the coloration, the shape, the color may be vary according to the fresh species, fresh fish that captured from aquatic body, and the preserved one, the color may be dull. Okay, this is the Channa Gachua. This is the Channa punctatus. You can see the coloration difference of fresh specimens. This is the Channa stratus. You can see the stripes on lower surface of the body. This is the Channa marulius. You can see the black spots and black blotches on lower surface of the body. Next one is the identification keys. Identification keys helps you to identify a particular type of fish species. Why I am saying keys like a lock key? Same, these key will help you directly to confirm what is the gender, what is the species. Okay, by following the method, you can do fish morphology also. If you are having a basic criteria how to identify a particular type of fish species, you can use the identification keys, you can use the standard literature in for fish identification. There are so many books are available. For example, Day Fauna is available, KC Jairam's books is also available. And these are generally for fish identification. Also, you can also see what type of morphometrical features they have shown in their book. There may be another book, so fishes of UP and Bihar, that is specifically for from the fauna, fish fauna of UP and Bihar. Okay, now I'm going to start the identification keys for actinopterygii. So there you can see there is a numbering on left side of the all points, and there may be a particular mentioning on a right side of all mentioned. So starting from the first point, that the head prolonged into a beak. If a fish is showing this type of feature, head prolonged into a beak, that it belongs to Balloniforms. If the head not belong to into a beak, that you can go to the point 2. Then the point 2 will be the body ill like. Okay, if body is ill like, then you have to go on third point. It means dorsal, you can have to check dorsal fin with the spines. If it is with the spines, that it is confirmed that it belongs to Metachembali forms. If dorsal fin without spines, then you can go to the next order. Okay, now taking the second example of second point, body is not eel like. If your fish for which you are doing morphometry, ish body is not eel like, then you have to go on point number 4, that is scales on the head. If scales on the head is present, then you have to go on point number 5th. If no scale is on head, you have to go on point number 7. So the point number 5 is dorsal fin with spine. Then you must see the sixth number of point, that is the spiny dorsal with 4 dorsal spines, ventral, abdominal, and subabdominal. Then this belongs to Muciliforms. Okay. If it is not, then you have to move on next point. So once you are having your identification keys and your fish with you for which you are doing the morphometry, then you have to check all these features, and these key will help you to go one by one to the next level. So when I'm searching for next nine number point, you can move by step by using this point. If you are searching for 7 point, a distinct ventral floor if present, then tetraodontiforms, it will go to the tetraodontiforms. It no float, then you have to go on ninth number of point. For ninth number of point, you must check abdominal keeled or sedated. If it is keeled, it or sedated and sedated, sorry, it will belong to order cryptiforms. If abdominal is not keeled, not round, or when keeled, that is not serrated, then it belongs to cypriniforms. So by using do, you are going to the particular type of order. So next you have to check the order for cypriniforms. So cypriniform deals with this work. For example, you have to check body covered with the scale, then it will belongs to cyprini. If not covered with scale, that it will be of silurini. That I have initially mentioned on the fishes, that I was showing the morphology, that a scaly fish, a non-scaly fish. Doesn't mean that non-scaly fish does not have any type of scale, but scales are embedded in the skin. But in case of cyprini, these will be appear on outside of the body. Okay, then you have to check a for cyprini for further features. Then you have to check scales are distinct, large, and prominent. Then you have to go cyprinidae family. If the scales are minute, rudimentary, embedded in the skin, and concealed under the mucus, then it will belong to Corbitary. Then you have to check the further feature of Cyprini. Then you must take the first feature apart of or whole of the abdominal edge keeled but not serrated. Then you have to go point 2. Then point 2 is the prominent type of showing prominent type of feature, peridorsal scales extended to the anterior orbital space, lateral lines gently curved downward above the pectorals, a knob at the symphysis of lower jaw which first into the corresponding depression upper jaw. Then it will belong to the genera Oxygaster. So by using the same features, you are going to the particular gender. Okay, by using these keys and these methods of identification, you can go to particular type of gender. And if you are going on particular type of gender, for example, you have reached on Serenus and the Livio, and it is confirmed that this particular fish belongs to this particular gender, then further you can identify these features. Okay, these are the identification keys and so on. So after the generic identification, the further features will be mentioned on these keys, and you can use the fin formula, so you can identify the particular fish at a species level. For example, there are so many features: Copuntius, Katla, Andras, Bora, Aspidoperia, Danio, Beryllus, and so on. So now I'm talking about the importance of fish taxonomy. There are so many various types of phenomenon present in the nature, but while we are dealing with this traditional feature, fish morphology, fish taxonomy, then this is an important evolutionary aspect for ichthyology. For example, if any fish type of fish species showing any morphological variation, then you can check, cross-check, verify, then catalog the particular type of species. Then you can also do the molecular identification, then molecular taxonomy, molecular phylogenetic. If on molecular phylogeny, it is closely related to any other type of fish species, then you can say the my species is directly closely related to the previous one, and this is the particular type of species that I have mentioned. Okay, if on the morphology it is showing some distinctive feature from previous one, and it is also showing some variation on the basis of molecular data, then you can say this species is totally different or somewhere different from previously described species. Though maybe you can resolve taxonomic ambiguity, maybe you can resolve any uh, taxonomic issues like any cryptic species. You can also describe any cryptic species, or maybe in same cases you can identify new species. So same will be used in cataloging, revision, handbooks, and also preparation of monograph. Then exotic species, maybe sometimes you have seen in nature there are so many hybrid species, there are so many intermediate forms of fishes are available in case of local market, where you can identify what is the natural threat and nature threat to our species, our local fish fauna, to our indigenous species. You can also identify in the identification of fishes or native fishes. So these are the basic importance in fish taxonomy. That is also a fish morphometrics. There are so many features on the basis of caudal fins, pectoral fins. There are shape, size, structure, coloration variations. So we will discuss the same in next lecture. So these are the basic morphological features for fish identification. These are the references those I wish to cite. The day fauna FO sheet, identification sheet. Also I have used their arms and Talwar and Jigran's books. These all are the good standard literature you can use for fish identification. At last, I would like to thanks to all of you for listening to me.